Hello and welcome to my Science Baccalaureate presentation about the effects of microplastics on humans and the solutions to microplastic pollution. I first became interested in this topic due to my aspiration to pursue a career in marine biology, with microplastics quickly becoming one of humans and the ocean's largest problems. What is a microplastic? A microplastic is a plastic particle between 5 mm and 0.1 micrometers. Microplastics are released into the environment when larger plastics degrade and when microplastics such as microbeads drain into the ocean. Scale of the problem. A researcher I had contact with called Garth Covington from the University of Victoria was involved in a study that estimated that the average American is consuming 70,000 microplastic particles per year through diet and inhalation. This number is quite frankly enormous. In 2019, it was also estimated in The Guardian that the global cost of the plastic pollution crisis was $2.5 trillion. However, by 2050, if current trends continue, there will be more plastic in the ocean by weight than fish. How plastics break down. In the natural environment, plastics will break into smaller and smaller pieces, and this process is aided by two mechanisms. The first of these is photodegradation, and this is when plastics absorb UV radiation from the sun, which excites electrons in order to form free radicals. These free radicals are very reactive and will react with elements such as oxygen in the air in order to break down the polymer. The other is wave action, and this is when the motion of the sea breaks down plastics into smaller fragments. Trophic transfer. This is one of the mechanisms in which microplastics can work their way up the food chain and into humans. Trophic transfer is when we eat other organisms that have previously consumed microplastic, transferring these microplastics into our own bodies. Through this pathway, humans could be consuming microplastics previously consumed by zooplankton, and as you can see in this video here, microplastics are being actively consumed by zooplankton. Most microplastic particles are expected to pass through the digestive system of the organism and be excreted unless they are small enough that they can be absorbed into the blood or are too large and get stuck in the digestive tract. Some foods are much higher risk than others for trophic transfer to humans, as when we eat organisms such as fish, the digestive tract of the organism is removed before the fish is consumed, meaning the area with the highest microplastic concentration is not consumed by humans. However, when crustaceans, like prawns, are consumed, the digestive system of the organism isn't removed, meaning the area with the highest microplastic concentration is consumed by humans, posing a greater risk of microplastic trophic transfer. Microplastics can also be consumed by humans when ingesting food or drinks previously contained in plastics, as some of this plastic will degrade and leach into our food and drinks before they are consumed. In the next part of my presentation, I will focus on the effects of microplastics on humans. Research from the University of Utrecht suggests that immune cells are recognizing microplastics in the blood. The effects of these microplastics on immune cells or white blood cells were tested by these researchers. Immune cells were isolated with or without microplastics, and in the cells isolated without microplastics, only 20% of all immune cells died in this time frame. However, in the immune cells isolated with microplastics, 60% of all immune cells died in the same period of time, illustrating the toxic impact that these microplastics could be having on our immune system. When exposed to microplastics, our immune cells attempt to destroy them, with immune cells like phagocytes attempt to engulf them, as seen here. This, however, causes the cell to die as a result of this exposure. Burning plastic waste produces dioxins, and halogenated plastics in particular are most concerned for producing these dioxins. An example of these halogenated plastics is PVC, which is an everyday plastic used in wire coatings and in pipes. Burning these plastics such as PVC produces polychlorinated dibenzodioxins or PCDDs, which is a group of persistent organic pollutants. When we inhale these PCDDs, they will dissolve in our adipose tissues or fats and will remain inside our bodies for an extended period of time, with most dioxins having a theorized half-life of 7 to 12 years, meaning they will still be inside our bodies for years to come, just in progressively smaller doses. The worst of these PCDDs is TCDD, as you can see in the bottom right. TCDD was a contaminant of the US weapon of war Agent Orange, which was sprayed over Vietnam during the Vietnam War. This particular dioxin causes birth defects, as when exposed to an embryo, it causes errors in cell division. During mitosis, or cell division, we start out with a single centrosome as seen here. This centrosome builds up and duplicates to form two separate centrosomes. TCDD causes errors in this duplication process, leading to damaged centrosomes pulling our chromosomes apart during mitosis, as the centrosomes control the movement of the chromosomes. This can damage DNA, as our chromosomes will be pulled apart in an incorrect fashion. This damaged DNA will then go on to cause birth defects, as all of the cell's offspring will inherit this damaged DNA, which will lead to altered genes being expressed, leading to visible differences in the child. In Vietnam, 150,000 children were born with birth defects as a result of exposure to this dioxin. Carcinogens. Plastic components such as formaldehyde are listed as carcinogenic. A carcinogen is any substance that promotes the formation of cancer, and it does this through promoting increased cellular division, which leads to an increased chance of abnormal DNA synthesis. This can lead to the formation of cancer cells. Formaldehyde is a component of the plastic Bakelite, and this is a very common plastic used in a huge variety of things from bake pads to kitchen containers. If these Bakelite microplastics enter our bodies, as these microplastics degrade, then formaldehyde molecules could possibly be released, which could lead to the formation of cancers. Additives to plastic. Certain chemicals are added to plastic in order to make the polymer stronger, more flexible, and more durable. The additive I will focus on is BPA, as you can see in the top right corner. This chemical is an endocrine disruptive chemical, or EDC, meaning it affects our hormonal system. BPA could leach out of microplastics when accidentally ingested by humans. BPA, or bisphenol A, is an EDC that was once present in baby bottles until it was banned in the EU and USA from being used in children's products. This is because BPA was leaching from these products and into the child. BPA is, however, still present in food packaging. BPA is a xenoestrogen, meaning it mimics the effects of estrogen on the human body and is an agonist for estrogen receptors. BPA therefore binds to estrogen receptors in the mammary gland, which causes division of mammary cells in the breasts. This division is uncontrolled, and excessive division could lead to the formation of cancer cells and tumours, causing breast cancer. BPA can also result in insulin resistance in women. This is because a high BPA concentration can lead to a similar set of circumstances arising that occurred in pregnancy when there is a high estrogen concentration. This is when the female body is desensitized to insulin temporarily in order for glucose to be diverted towards the embryo and not to be absorbed by the mother. However, in this case, there is no embryo to divert this glucose towards, meaning a high glucose concentration is left circulating in the blood. This gives an increased risk of atheromas or plaques forming in the blood when this glucose clumps together. This can lead to atherosclerosis, which is a common disease for people with type 2 diabetes, and is when our arteries become clogged by these atheromas, limiting blood flow. BPA was also found to disrupt the gene expression of KCC2. KCC2 is a chloride ion transporter protein that pumps chloride ions out of a neuron. Chloride ions are in a particularly high concentration in developing neurons, and the dysfunction of this chloride ion transporter protein can result in neurological disorders. BPA has also been seen to bind to androgen receptors, and this is the receptor for hormones such as testosterone, and BPA is an antagonist for this receptor, meaning it binds to this receptor but does not stimulate a response. This leads to testosterone having no effect as its receptor is occupied by the BPA molecule. This can then result in males having a low sperm count as testosterone cannot stimulate sperm production. Plastic vectors. A vector is something that is used as a vehicle to carry foreign material from one place to another. Microplastic's ability to carry hazardous pollutants was tested by researchers from the Autonomous University of Madrid and Alcalá University using the antimicrobial triclosan, or TCS. The microplastics that best sorbed the TCS were exposed to freshwater algae, and significant decreases in growth of the algae and chlorophyll A content were observed. This evidence suggests that microplastics can carry harmful materials and release them when the correct conditions arise. This does not simply just apply to hazardous chemicals, however, as pathogenic bacteria such as Vibrio parahemolyticus have been seen to hitchhike on ocean plastic. This pathogen causes gastrointestinal illness in humans. Therefore, if humans accidentally consume microplastic particles, we could be transferring this pathogen or others into our own bodies, leading to disease. Microplastics acting as a vector implies that it is not simply just the microplastic particles that can cause disease in humans. Disease can also be caused by the harmful chemicals and pathogens these microplastics could be transferring into our own bodies. To summarise the effects on humans section of my presentation, so far we've looked at the toxic impact of microplastics on the immune system. Babies having birth defects is the result of exposure to the dioxins produced when burning plastic waste. The components and additives to plastic and how these can cause cancers as microplastics degrade. How the additive BPA affects our hormonal system. And how microplastics can act as a vector for harmful substances and pathogens. The only way to prevent these profound impacts on humans is to not allow these microplastics to enter our bodies by cleaning up their microplastic pollution. This can be done in a variety of ways which I will now discuss. Ocean cleanup. This was a project designed to try and tackle the huge collection of marine waste that is gathered at the centre of the Pacific Ocean. This is an area known as the Great Pacific Garbage Patch, and they were going to do this in order to tackle much larger marine waste before it degraded further into harder to clear up microplastics. They were going to try and tackle this problem through the creation of huge booms which would traverse the ocean and gather plastic waste on the surface as they moved. However, this project had huge limitations as the booms simply moved far too slowly and plastic waste floated away from them. Biodegradable plastic. The most common bioplastic is PLA or polylactic acid and is made from cornstarch instead of fossil fuels. The cornstarch molecules are fermented in order to produce lactic acid as seen in the diagram. This lactic acid is then polymerized to produce polylactic acid or PLA. This bioplastic can then be broken down in nature as the PLA monomer units will slowly absorb water and break into smaller fragments. These can then be digested by many microorganisms to produce CO2 and water. However, many bioplastics currently produced will not break down in nature like PLA does, adding to our microplastic problem. But despite this, biodegradable plastics like PLA are a realistic solution to our excessive usage of single-use plastics, as they involve neither chopping down of trees like paper or crude oil. I believe the overall solution to microplastic pollution is the evolution of organisms in order to digest plastics. The first of these creatures is the wax moth caterpillar Baccalaureate melanella, which has been seen to digest polyethylene. It can do this as it's in a mutualistic relationship with bacteria in its gut microbiome, which were aiding it in the breakdown of polyethylene. This is important as it suggests other organisms could evolve in a similar way, being in a relationship with microorganisms which can aid them in digesting plastics. Alicia Matthews Cardenas is a researcher at Cork University which I was in contact with. She was involved in the discovery of another organism called Gammarus duodeni, which has been seen to rapidly fragment microplastics. This freshwater amphipod breaks down microplastics into much smaller nanoplastics as they travel through its digestive system. Ideonella sakaiensis 201F6. This was the first bacterium to be discovered that was digesting plastic and was surviving by breaking down the plastic PET or polyethylene terephthalate. This is a very common everyday plastic used in materials such as plastic bottles. This bacterium was discovered by researchers at the Kyoto Institute of Technology and the Keio University, which are both in Japan. This bacterium can break down PET as it has two revolutionary enzymes. The first of these is PETase, which is what you can see here. This PETase breaks down the PET into BHET, MHET, and TPA. And then there's a second enzyme working in tandem with the first, called MHETase. This breaks down MHET into TPA or terephthalic acid and EG or ethylene glycol. These two products can then be used by the organism in order to survive on PET as its primary carbon source. For many years, PET was believed to be an indestructible material in the environment, but it can now be broken down completely by bacteria, demonstrating the incredible rate at which these microorganisms are evolving to their changing environments. Engineered plastic enzymes. Scientists from the University of Portsmouth and the National Renewable Energy Laboratory USA altered some of the amino acids on the active site of the aforementioned PETase. This alteration created a mutant version of the enzyme that had a greater affinity for PET, leading to a 20% increase in the productivity of the enzyme. This is important for the future as it conveys that these naturally produced enzymes are imperfect and can be further optimized for the shape of their substrate, which could lead to this plastic breakdown becoming more and more effective, and could lead to new advancements in fields such as recycling as this plastic breakdown will become much faster. Looking to the future. New waste collection systems are currently being produced by companies such as Ocean Cleanup in order to target plastic waste at its source, which is in the world's largest rivers. In the future, I also believe more organisms will evolve in order to break down different types of microplastics circulating in the ocean, similarly to the organisms I have discussed today have. New technological advancements will also play a huge role in solving the microplastic problem, such as implementing the PETase gene into other organisms such as E. coli. Humans through this method could increase the number of organisms that can biodegrade plastics like PET, instead of having to wait for these organisms to adapt to their changing environment. Thank you for listening. I'd like to thank the following people for helping me produce this presentation. I'd like to thank Karen Hall, Alicia Matthews Cardenas, Garth Covington, and especially my baccalaureate tutors, Mrs. Reed and Mr. McGinley.